Good morning, student. In this video, I shall discuss the chromosomal theory of inheritance, which was actually given by W. Shorten and Boveri in 1902. Although Mendel conducted his famous experiment in around 1856 and he published in 1865, but till 1900 his work was unrecognized due to certain reasons, because at that time no much communication facilities were available. So that is why, although it was published in a journal, but it was not communicated to the biologists actually who were working on them. Second, he actually given the idea of unit character, that means the factors which are unit and non-blending nature. That is why these were actually not accepted by the contemporary scientists like Darwin, Wallace, etc. because they were mainly in habituation or in acceptance of the blending nature which were normally environmental influence. So that is why these are the main factors for which it was not recognized. Another important factor was that the mathematical use or mathematical and statistical use in the experiment or in the analysis of the data of the experiment. And most of the biologists, they were not acquainted with these mathematics and statistical tools. So that is why it was not recognized till 1900, and Hugo de Vries, Carens and Tesserma, these three scientists, they recognized them around 1900 AD. But side by side, some other scientists were working on the cell and they already discovered the chromosome, and Shorten and Boveri, they also explained the behavior of the chromosome during gamete formation. And the Mendel and they observed that the behavior of the chromosomes and the behavior of the factors as explained by Mendel, both are more or less similar. Now how? The chromosomes actually behave during the gamete formation. During gamete formation, as we know, the mother cell, normally either the sperm mother cell or egg mother cell, it is diploid in nature. These are the somatic cells, and these diploid cells, when they form the gametes are normally haploid in nature. Then how these chromosomes behave during this process of meiosis, you have already studied in class 11. Now here, suppose there are 4 chromosomes in the somatic diploid cell, in the sperm mother cell or the egg mother cell. Now 1 and 2, suppose these are homologous chromosomes. Then what is the homologous? What are homologous chromosomes? The chromosomes which are present in a particular organism. Suppose one organism is here, and whatever chromosomes which are present here, one set is coming from the father, another set coming from the mother. So in case of human being, there are 23 chromosomes coming from father and 23 chromosomes coming from mother. And these 23 chromosomes, these are the haploid set of chromosomes in the form of the gamete. And when they present there in the offspring, there are 23 pairs of chromosomes. Now these 23 pairs of chromosomes are actually one set 1 to 23 from father and 1 to 23 from mother. So that is why chromosome number 1 of father and chromosome number 1 of mother, these two chromosomes are called the homologous chromosomes, or these are the homologous pairs. So each diploid organism, whatever chromosomes are there, they are diploid in nature. That means one from father side or paternal side and another from maternal side. So these two are combinedly called the homologous pair. Now these homologous chromosomes, they normally pair with one another. And why they pair with one another? Because the crossing over occur. During crossing over, some sort of exchange of segment occurs between the non-sister chromatids. The chromatids of one chromosome are called the sister chromatid, and the chromatids of other chromosome is called the non-sister chromatid. Now the exchange of segment occurs. After that, they again separated from one another after exchange. So during meiosis 1, these events occur. Now look at the chromosome. Suppose chromosome number 1, 2, 3, 4. 4 chromosomes are here. Out of these, 1 and 2 are homologous. Suppose here, chromosome number 1 and 2, these are homologous, and chromosome number 3 and 4, these are homologous chromosomes. So these homologous chromosomes, they are normally bivalent or paired together. Chromosome 1 and 2, they paired together here. So here in case of meiosis 1, they pair together and the exchange of the segment occurs by process of crossing over. And after that 3, 4, they are homologous. That is why they also combine together to form the bivalent. Now after that the two cells are formed after meiosis 1, and one is having one set of chromosomes, they are separated from one another, and one set goes here, another set goes here. So 1 and 3, chromosome number 1 and 3 goes to here, and chromosome number 2 and 4, because 1 and 2 are homologous and 3 and 4 are homologous. So that is why they are separated from one another and they go to different cells. But in meiosis 2, what happens? Now these two again split because this division is equivalent to the mitosis type. So splitting of the centromere occurs here, and one chromatid goes to this gamete, another chromatid goes to this gamete, another chromatid from this cell to this and this. And there are 4 daughter cells which are formed here, and each daughter cell having haploid number of chromosomes. Suppose here the 4 chromosomes were there in the mother cell, but here you will find 2 chromosomes are there in each cell. So that is why these are called the haploid cell. So this type of behavior of the chromosomes occur during the gamete formation or during the meiosis process. Now this process is very much important for the formation of the gamete. And now after that they compare the behavior of this chromosome with the behavior of the factors or the genes which were explained by Mendel. Now the behavior of chromosome during the independent assortment of the different alleles which are present in the different chromosomes. Now here according to Mendel, the independent assortment of the pair of chromosomes, or you can say the pair of factor, or here we can explain it in the form of the chromosomes. Now in the mother cell of any germ cell, or you can say any gamete formation cell, this is either the sperm mother cell, or it is the egg mother cell, or in case of microspore, into microspore mother cell or megaspore mother cell. And these mother cells, they are normally diploid in nature, having 2 number of chromosomes, and these chromosomes are actually homologous in nature because one set coming from the father and one from mother. So that is why suppose here chromosome number 1 and 2, these are homologous chromosomes, and chromosome number 3 and 4 are also homologous chromosomes. Of course, these chromosomes, they contain the dominant recessive genes, that is a separate thing. But here, these homologous chromosomes, each chromosome having, suppose this is one chromosome, so this chromosome having 2 chromatids, these are called chromatids, and each chromosome consists of 2 chromatids. And these chromatids normally they separate from one another during gamete formation. So that is why suppose these chromatids are there in a particular chromosome, these chromosomes are now separated during the meiosis, or this is called the meiosis 1 and then the meiosis 2, and during this meiosis 2 how the pairing of the homologous chromosome occurs. Okay, so during this pairing and the separation, what happened? They split from one another, and normally look at here how they split from one another. They are normally present 1, 2, 3, 4, but they 1 with 3 and 2 with 4 comes here. Now during this independent assortment of the chromosomes, or you can say during meiosis, during gamete formation, how the chromosomes, they are independently assorted or independently they are transferred to the gamete cell, and from the gamete cell they are transferred to the next generation. Now this is independentness of these chromosomes are in this manner, just like the factors as explained by Mendel. Now here 4 chromosomes are there, 1, 2, 3, 4. 1 and 2 are homologous chromosomes and 3 and 4 are homologous chromosomes. So that is why these homologous chromosomes, they pair together and they again separated together because during this phase the crossing over occur, and during crossing over the exchange of segment occurs. So whatever may be it occurs, but after that they are separated from one another, and one or this one half of the pair, because these are homologous pair, that means this must be goes to one cell and this must be goes to another cell. So this half, this is 1 and 3, they come here, and 2 and 4 they come here, and also here 1 and 4, look at here, this is the random separation. There is no link of this chromosome with this or this chromosome with this, but if they randomly, now 1 with 4 they remain together, and when this cell was splitted or separated, now 1 and 4 goes to one cell and another 2 and 3 goes to another cell. Okay, so that is why what happened, look at here, now this chromosome number 1 and chromosome number 3 they are present here, and chromosome number 2 and 4 here, and 1 and 4 and 2 and 3 here. Now they again in meiosis 2, which is similar to the mitosis, they split and their chromatids actually split here. But during this meiosis 1, normally they just 2 chromosomes, they reunited and they separated. So that is why there is no splitting of chromatid occur, but here what happened, the splitting of chromatid from the centromere occur. Now each chromatid of the chromosome, now they move to one gamete. Now here one chromatid of chromosome 1 come here, another from chromatid 1 is here, and chromatid 3 one part here, and another chromatid 3 here. So in this way they split and they are independently moved to the different gamete, and that is why there are different 4 types of cells you will find here, 1 4 1 4 and 2 3 2 3, here 1 3 1 3 and 2 4 2 4. So that is why these are the different types of combinations of the gamete which are formed in the offspring. So that is why they will form the permutation combination method, and the different types of zygotes are formed and they are randomly formed. So that is why no chromosome is normally linked with one another, they are independent with one another, and that is why they are asserted to the next generation independently. And this phenomenon, which was explained by Mendel in his factor, and the same phenomenon is also observed in the chromosomes. So that is why the behavior of the chromosomes, as explained by Sutton and Boveri, and the behavior of the factors, as explained by Mendel, both are similar. Now, when we compare the behavior of the chromosome and the behavior of the genes, the behavior of the chromosome was actually explained by Sutton and Boveri, and the gene, or you can say this is the factor as explained by the Mendel. And according to Mendel, the factors like your T or T or capital T small t, so these are the two alleles and they always occur in pairs. So normally they occur in pairs. Like that, in case of chromosomes, here Sutton and Boveri also explained that the chromosomes always occurs in pairs. How? So in case of human being, there are 23 pairs of chromosomes. Why not 46? Because there are actually 46 chromosomes, but out of these 46, 23 comes from the father side or paternal side and 23 comes from the mother side. So that is why the chromosome number 1 of father and chromosome number 1 of mother, this is one pair. Chromosome number 2 of father and chromosome number 2 of mother, these are another pair. Like that, there are up to 23 and up to 23. Except XY, all are homologous chromosomes. And that is why they also occur in pairs. So that is why there is a very good similarity between the chromosomes and the genes. Second, now they normally segregate during gamete formation. During gamete formation, and during segregation, the chromosomes they segregate in such a way that the one chromosome, suppose this is one of the homologous chromosome and it is another of the homologous chromosome, and these two they actually split from one another, and one of those homologous pair comes here and another of this homologous pair goes here. So never these two go to one gamete. So that is why this is called segregation of the chromosomes during gamete formation, so that one set of chromosomes goes to one gamete and another set straight goes to another gamete. So this type of segregation also occurs here in case of factors on genes segregation, and here Mendel explained how this, suppose this is one pair in the F1 generation monohybrid cross, and during gamete formation one gamete is formed like this, another gamete is formed like this. So that is why these are segregated from one another to form the gamete, or during gamete formation. This is one gamete and this is another gamete. So both case, in case of chromosomes as well as in case of genes, they segregate during gamete formation, and after that, next, how they segregate? Now, third one, that is the independently. That is called independently. They are segregated and reunited, so that the combinations are always randomly occur, and this random or independent assortment of the chromosome occurs here in case of chromosomal theory explained by Sutton and Boveri, and this independent assortment of the pair of genes or factors occur, and they are independent of another factor. Suppose it is round and it is yellow. So here these factors which are here and here, they are independent of one another. So that is why they are randomly segregated and they are randomly reunited, so that new combinations are actually observed here. So that is why there is a great similarity between the genes or the factors as explained by the Mendel and the chromosomes as explained by Sutton and Boveri. So that is why this is very good example for the chromosome and the genes and great similarity. Of course, there are some differences, later on it was already discovered, but still then this behavior of chromosome and behavior of gene are more or less same, and this is called the chromosomal behavior of inheritance as explained by Sutton and Boveri. Now, I hope this video will be useful for you, and if it is really useful for you, then please like it and subscribe for further videos. Thank you again.